Dear viewers, hello and welcome to another session of our lecture series. Today we will discuss an important topic which is entitled as Structure, Types and Functions of Lipids. Lipids, as we know, are a diverse group of organic non-polar compounds which are soluble in organic solvents such as chloroform and insoluble in hydrophilic or polar solvents like water. Lipids form one of the fundamental components of cell structure and perform vital biological functions. As structural components, they play a crucial role in membrane structure and synthesis by being the major constituents of the cell membranes. They are also cited as the important energy storage molecules. Based on their structure, the lipids are classified into four types: simple lipids, compound or complex lipids, derived lipids and miscellaneous lipids. Let's first talk about the simple lipids. Simple lipids are the lipids which are composed of fatty acids and various types of alcohols which are linked together by ester linkages. The simple lipids are of two types: triglycerides and waxes. So far as the triglycerols are concerned, these lipids are the esters of fatty acid and trihydric alcohol glycerol, where the three hydroxyl groups of glycerol are engaged in ester bonds with three fatty acid molecules. These lipids are thus called as triglycerides. They are also commonly known as neutral fats. Depending upon the type of fatty acids joined to the three hydroxyl groups in glycerol, the triglycerides are of two types: simple triglycerides and mixed triglycerides. Simple triglycerides are those triglycerides in which the three hydroxyl groups of glycerol are bonded to three same types of fatty acids. Tristearine is the best example of simple triglycerides, where the three hydroxyl groups of glycerol are bonded to three molecules of stearic acid. In triolene, the fatty acid is oleic acid, which is joined to glycerol, while in tripalmitin, the fatty acid is palmitic acid. On the other hand, if more than one type of fatty acid is esterified with trihydric glycerol, the resultant triglyceride is called as mixed triglyceride. In other words, in mixed triglycerides, two or three different kinds of fatty acids are esterified with glycerol. In nature, only few triglycerides are of the simple type, and most of the triglycerides are thus of the mixed triglyceride type. Sterodipalmitin is an example of mixed triglycerides in which two palmitic acid residues and one stearic acid is joined with the glycerol by ester linkages. In oleopalmitostearine, which is also a mixed triglyceride, the three hydroxyl groups of glycerol are bonded with three different kinds of fatty acids, which are oleic acid, palmitic acid and stearic acid. The fatty acids constitute about 94 to 96% of the total weight of triglycerides. They influence many physical and chemical properties of triglycerides. The triglycerides which have only saturated fatty acids, that is the acids having only single bonds between their carbon atoms, are solid at room temperature, while as the ones which have unsaturated fatty acids, that is acids with one or more double bonds in them, are liquid at room temperature. The example of solid triglycerides is the animal fat, while as soybean oil is the example of liquid triglycerides. After triglycerides, we now come to the second type of simple lipids, that is the waxes. Waxes are esters of long chain monohydric alcohols and fatty acids. The long chain monohydric alcohols in waxes have 16 to 36 carbon atoms, while as the fatty acids range between carbon 14 to carbon 36. The examples of waxes include bee wax, spermaceti wax, carnauba wax and lanolin. The waxes from the conifers contain polymers which are formed by the ester linking between many omega hydroxy acids such as juniperic acid. The second class or the category of lipids is the compound lipids. The lipids which have parts other than or including fatty acids and glycerol are called as compound lipids. These lipids may have, in addition to glycerol and fatty acids, some other structures such as sugars, phosphorus and amino alcohols conjugated in their structure. The compound lipids are also called as complex lipids or conjugate lipids. There are two common types, one phospholipids and two sphingolipids. The phospholipids or glycerophospholipids are the structural lipids which have a glycerol backbone with two hydroxyl groups joined with two long chain fatty acids and the third hydroxyl group attached to a phosphoric acid molecule which is conjugated on the other side to some other molecule such as an alcohol, amino acid or a sugar molecule. The phospholipids are the major class of lipids which enter into the structure of biological membranes. The phospholipids are amphipathic with glycerol and phosphate forming the polar or hydrophilic end of the molecule, while as the hydrocarbon chains of fatty acids forming the non-polar part of the molecule. In phospholipids, the two different fatty acids attached at carbon 1 and carbon 2 of glycerol differ from one phospholipid molecule to another, besides a variant head group attached to phosphate, thus resulting in about 1,000 different possible types of phospholipids. In phospholipids, usually the fatty acid attached at carbon 1 of glycerol is a saturated fatty acid, while as the fatty acid attached at carbon 2 is usually an unsaturated one. The two common phospholipids found in biological membranes are lecithin and cephalin. In lecithin, the alcohol linked to the phosphate joined to carbon 3 of glycerol is choline. Thus, lecithin is also named as phosphatidylcholine. In cephalin, the alcohol attached to phosphate is ethanolamine. Thus, cephalin is phosphatidylethanolamine. These are also called as phosphatides. After discussing phospholipids, we will now talk about the sphingolipids. Sphingolipids is one more class of lipids which enter into the structure of cell membranes. These lipids are those compound lipids where glycerol is replaced by a long chain base sphingosine, which is linked by an amide bond with a fatty acid. This combination of a sphingosine with a fatty acid by amide bond is called the ceramide unit. Since all sphingolipids contain a common ceramide unit, it is the varied head group attached to the sphingosine which accounts for the different types of sphingolipids. The common sphingolipids are sphingomyelin, glycolipids or cerebrosides, gangliosides. The first type, that is sphingomyelin, is a sphingolipid comprising of a ceramide unit and phosphorylcholine. In sphingomyelin, the phosphorylcholine is joined to hydroxyl group of ceramide at position 1. Sphingomyelin is found as a major component of the complex lipids in animal tissues, but not in plants and microorganisms. The second type, that is cerebrosides, consists of a ceramide unit bonded to either glucose or a galactose molecule. Based on the sugar moiety bonded to the ceramide, the two types of cerebrosides are glucocerebrosides and galactocerebrosides. The third and last type of sphingolipids are the gangliosides. These gangliosides are those sphingolipids in which a ceramide is joined to an oligosaccharide having one or more sialic acids attached to its sugar chain. Dear viewers, we now come to the third class or category of lipids, that is the derived lipids. Derived lipids is another class of lipids and includes those lipids which are obtained from simple and complex lipids on their hydrolysis. These include fatty acids, alcohol, monoacylglycerides, diacylglycerides, prostaglandins, lipid-soluble vitamins, steroid hormones, etc. A fatty acid is an organic acid with a long hydrocarbon chain which ends in a carboxylic group. The carbon chain length in the fatty acids varies from 14 to 22 carbon atoms, with most of the fatty acids having 16 to 18 carbon atoms in their structure. Most of the fatty acids have an even number of carbon atoms as they are synthesized from 2 carbon acetyl coenzyme A molecules, while as there are also fatty acids such as propionic acid and pentanoic acid which have an odd number of carbon atoms constituting them. A fatty acid without any carbon-carbon double bond in its hydrocarbon chain is called as a saturated fatty acid. For example, palmitic acid and stearic acid. While as fatty acids which have carbon-carbon double bond in their molecular chain are called as unsaturated fatty acids. A fatty acid with single double bond is called as monounsaturated fatty acid, while as polyunsaturated fatty acids have more than one double bonds in their structure. Glycerol, which is an important constituent of the lipids, is a trihedric alcohol having 3 hydroxyl groups which can be bonded to 3 fatty acids. When one fatty acid is bonded with one of the hydroxyl groups of glycerol, the resulting compound is a monoglyceride. And if 2 fatty acids are bonded with 2 hydroxyl groups of glycerol, the compound is a diglyceride. Triglycerides are formed when all the 3 hydroxyl groups of glycerol are engaged in bond formation with 3 fatty acids. Monoglycerides and diglycerides are partial esters of glycerol having 1 and 2 fatty acids respectively. They do not occur in natural fats but are produced on partial hydrolysis of triglycerides. Let's now know what are the steroids. Steroids are the lipids which are characterized by a fused 4-member ring structure comprising 17 carbon atoms. The core of steroids has 3 cyclohexane rings and a 4th cyclopentane ring, also called as gonane structure. Generally, steroids have a methyl group attached at carbon 10 and carbon 13 and a side chain or alkyl group attached at carbon number 17. Depending upon the functional groups attached to the fused ring structure, the steroids are of many types which include sterols, cholesterol, bile acids and steroid hormones such as corticosterone and androgens or sex hormones. The sterols are the steroids that have a hydroxyl group attached at carbon 3. They are thus also called as steroid alcohols. The sterols occur naturally in plants, animals and fungi. The sterols occurring in plants are called as phytosterols and those of the animal origin are called as zoosterols. The most common sterol found in animal world is the cholesterol, which is encountered in the architecture of cell membranes where it lends rigidity to the cell membrane. The common phytosterols include stigmasterol, campestrol and cytosterol. Ergosterol is the sterol which occurs in fungi where it is found in cell membranes. It also provides rigidity to the fungal cell membranes. Now we come to the last category of lipids, that is the miscellaneous lipids. These are the lipids which do not belong to any of the above 3 discussed classes and have characteristics of lipids. These include squalene, terpenes and carotenoids. The terpenes are hydrocarbons having carbon skeleton derived from isoprene. Thus, they are also called as isoprenoids. The terpenes are classified into monoterpenes with 10 carbon atoms, sesquiterpenes with 3 isoprene units or 15 carbon atoms, C20 diterpenes and C30 triterpenes. The carotenoids are tetraterpenes having 40 carbon atoms. Now, after knowing the structure and various classes of lipids, we now come to the last segment of our topic, that is the functions of lipids. The first function of lipids is the constitution of biological membranes. The lipids, especially phospholipids, glycolipids and steroids such as cholesterol, are the main structural components of cell membranes. The lipid bilayer of plasma membrane and other biomembranes is composed of a wide variety of phospholipids and other structural lipid elements. Thus, the compartmentalization in eukaryotic cells is brought about by membrane-bound organelles such as mitochondria, chloroplasts, peroxisomes, etc. that carry out different biological functions. The second function of these lipids is that they are the source of energy. The fatty acids of triglycerides are a rich source of energy and are thus stored in both animal and plant structures as triglycerides for energy storage compounds. The fatty acids on oxidation release more energy than any other biological fuel such as glucose. In many plants, food is stored in small space in seeds in the form of triglycerides, which are utilized during seed germination. These lipids also act as hormones. In animals, many lipids serve as chemical messengers which coordinate many cellular and physiological functions. The important lipid hormones include steroid hormones such as aldosterone and other sex hormones such as testosterone, progesterone and estrogen. Insect hormones such as pheromones are also lipidic in nature. Lipids are also vitamins. Many vitamins such as vitamin A, vitamin K, etc. are lipidic in nature. They serve as important coenzymes of many important enzymes which bring important metabolic reactions inside the cells. Another important role of lipids is in the formation of protective coverings. In plants, the wax, which is an important lipid, forms protective covering on many plant parts such as leaves, where it helps them to prevent water loss and reflect excessive light, thus protecting the plants from the bad effects of external physical factors. The carotenoids in plants are accessory photosynthetic pigments which help plants absorb solar energy and also protect the chlorophyll from photooxidation. And lastly, the volatile olfactory compounds emitted by plants for attracting pollinators are also lipidic in nature. Dear viewers, with this we come to the end of today's session. wherein we discussed in detail the structure and different types or classes of lipids. Hope that we were able to justify the title properly. Thank you.